Hi, my name is Ingrid, and my technique assigned was RT-PCR. So RT-PCR is known as reverse transcriptase PCR, and it is used to study gene expression, and sometimes results can be altered by DNA contamination. The principle of this technique is the isolation of RNA. RT-PCR makes a complementary DNA using a reverse transcriptase enzyme. It is amplified using a PCR and then gel electrophoresis for the results. This technique is applied to distinguish exons and introns. It can sometimes be combined with real-time PCR in order to quantify mRNA levels and also to diagnose genetic diseases as well. This is a figure from, this is a figure from the primary research article that I found, and the question that the authors were asking was if there was a difference between PCR and RT-PCR to detect alive and dead bacteria. And the reason they were asking this type of question was because they had noticed that PCR was giving false positives in regards to this type of research. And the experimental design that was used was a different concentration of bacteria, 16 sRNA and EFTU primers, and they were also using RT-PCR and PCR. The bacteria that was being used was E. coli, and bacterial RNA was extracted and purified. The deoxyribonuclease enzyme, there's deoxyribonuclease 1 enzyme was used to eliminate DNA contamination, and the EFTU primer was used in order to prevent false positives results to come up in regards to the PCRs. In this figure, it shows the gel of the RT-PCR and the regular PCR. The first lane is the marker, which is 100 base pairs, and lane 2 and 3 are from RT-PCR. Lane 2 shows the live bacteria, and lane 3 shows the dead bacteria. Lane 4 and 5 are from the regular PCR, and lane 4 is supposed to show the live bacteria, and lane 5 is showing the dead bacteria. The low stability of RNA being used outside of the cell makes RT-PCR great for detecting living cells, and the RT-PCR does show that it is good and very efficient in order to detect less stable EFTU primer, and the PCR is not able to discriminate between dead and alive bacteria, which results in giving a false positive. And if you see in lane 5, it shows like a result for dead bacteria, unlike in lane 3. So right there, it's giving a false positive because it's not supposed to show it. And one of the strengths that I thought for this experiment was the EFTU primer used. It was supposed to prevent false positives for the RT-PCR and PCR. Some of the drawbacks that I thought that was used in this research was that the different concentration of bacteria on each PCR maybe could have played a part on it as well. And my question in regards to this article or journal, they mentioned that PCR may give false positives or for certain results. And what other studies would PCR give false positives, and what could be some factors? And these are my references. And that concludes my presentation. Thank you.